In this video, we are going to learn about the extracellular matrix and cell interactions. So, although the plasma membrane constitutes the boundary between a living cell and its non-living environment, materials present outside the plasma membrane play an important role in the life of a cell. Most cells in a multicellular plant or animal are organized into mostly defined tissues in which the component cells maintain a definite relationship with one another and the extracellular material that lie between the cells. Even those cells that have no fixed relationship within a solid tissue, such as white blood cells that patrol the body, must interact in highly specific ways with other cells and with extracellular materials in which they come in contact. These interactions regulate such diverse activities as cell migration, cell growth, and cell differentiation, and they determine the three-dimensional organization of tissues and organs that emerges during embryonic development. We will focus in this session on the extracellular environment of cells and the type of interactions in which cells engage. Epithelial tissue is a very important part. That is the outer layer of skin, the dermis, is a type of epithelial tissue. Like other epithelia, which lines the spaces and free surfaces of the body, the epidermis consists largely of loosely packed cells attached to one another and to an underlying non-cellular layer by specialized contacts. These contacts provide a mechanism for cells to adhere to and communicate with one another. In contrast, the deeper layer of the skin, the dermis, is a type of connective tissue. So we have learned that epithelial tissue is there, and along with that connective tissue will be there. Like other connective tissues, such as those that form a tendon or cartilage, the dermis consists largely of extracellular material, including a variety of distinct fibers that interact with each other in specific ways. A closer look at one of the scattered cells or fibroblasts of the dermis are given. The cell surface receptors interact not only with the external surroundings but are contacted in their internal end to various cytoplasmic proteins. Receptors with this type of dual attachment are well suited to transmit messages between a cell and its environment. So, if we begin at the plasma membrane and move outward, we can examine the types of extracellular elements that surround various types of cells. It was noticed virtually all integral membrane proteins as well as certain membrane lipids bears chains of sugars or oligosaccharides of various length that project outward from the plasma membrane. These carbohydrate projections from part of a body and form part of a layer closely applied to the outer surface of the plasma membrane called the glycocalyx or cell coat. This extracellular material is very prominent in some types of cells, such as the epithelial cells that lie in the mammalian digestive tract. The glycocalyx is thought to mediate cell-cell and cell subtractive interactions, provide a mechanical protection to cells, serve as a barrier to particles moving toward the plasma membrane, and bind important regulatory factors that act on the cell surface. So when we just look at the structure of how cells are organized into tissues and how they interact with one another and with their extracellular environment, we can see dead cornified cells are there. Epidermis, dividing cells situated on the downside. There is a basement membrane along with that dermis. So when we zoom the portion of basement membrane and along with some dividing cells, we can see specialized cell-cell contact, specialized cell substratum contact along with that basement membrane. Then we just try to zoom the dermis portion. You can see that reticular fiber will be there, proteoglycans are there, collagen fiber, cell surface receptor, that is the integral type. Fibroblast is there and elastic fiber will be there. So these are the structures of a extracellular environment. Now, the extracellular matrix. Many types of animal cells are surrounded by an extracellular matrix or ECM. ECM is a short form of this extracellular matrix, and we will be using that term after many sentences in this video. So, an organized network of extracellular materials that are present beyond the immediate vicinity of the plasma membrane. The ECM is more than an inert packing material or a non-specific glue that holds cells together. It provides both physical and biochemical signals that can play regulatory roles in determining the shape and activities of the cell. For example, enzymatic digestion of the ECM that surrounds cultured cartilage cells or mammary gland epithelial cells cause a marked decrease in the synthesis and secretory activities of the cell. So that part is very important, and we culture it externally also. So addition of extracellular matrix materials back to the culture dish can restore the differentiated state of the cells and their ability to manufacture their usual cell products. One of the best defined extracellular matrix is the basement membrane or basal lamina, a continuous sheet 5 to 200 mm thick that surrounds nerve fibers, muscles, and fat cells, and it underlies the basal surface of epithelial tissue such as the epidermis of the skin, or lining the digestive and respiratory tracts, underlies the inner endothelial lining of blood vessels. Basement membrane provides mechanical support for the attached cells, generate signals that contain and maintain cell surface, serve as a substratum for cell migration, separate adjacent tissues within an organ, and act as a barrier to the passage of macromolecules. In their greater capacity, basement membranes of the capillaries play an important role in preventing the passage of proteins out of blood and into the tissues. This is particularly important at the kidney, whereas the blood is filtered under high pressure through a double-layered basement membrane that separates the capillaries of the glomerulus from the walls of the kidney tubule. Kidney failure and long-term diabetes may result from an abnormal thickening of the basement membranes surrounding the glomeruli. Basement membranes also serve as a barrier to inversion of tissues by cancer cells. The molecular organization of basement membrane is discussed later, and those cells that do not lie on a basement membrane sheet, such as the fibroblasts of a connective tissue, are typically surrounded by a less organized ECM consisting largely of thread-like fibrils. Even though the extracellular matrix may take diverse forms in different tissues and organisms, it tends to be composed of similar macromolecules. Unlike most proteins present inside the cells, which are compact globular molecules, those of the extracellular space are typically extended fiber species. These proteins are secreted into the extracellular space where they are capable of self-assembling. So, the extracellular space contain extended fibrous species. These proteins are secreted into the extracellular space where they are capable of self-assembling into an interconnected three-dimensional network, which is depicted. And among their diverse functions, the proteins of the ECM serve as trail markers, scaffolds, wires, and glue. As noted throughout the following sections, alterations in the amino acid sequence of extracellular proteins can lead to serious disorders. We will begin with one of the most important and ubiquitous molecules of the ECM, the glycoprotein collagen. Collagen comprises a family of fibrous glycoproteins that are present only in the extracellular matrix. Collagens are found throughout the animal kingdom and are noted for their high tensile strength, that is, their resistance to pulling forces. Collagen comprises a family of fibrous glycoproteins that are present only in the extracellular matrix. Collagens are found throughout the animal kingdom and are noted for their high tensile strength, that is, their resistance to pulling force. It is estimated that a collagen fiber of 1 mm in diameter is capable of suspending a weight of 10 kg without breaking. So that is an amazing property. Collagen is the single most abundant protein in the human body, constituting more than 25% of all protein, a fact that reflects the widespread occurrence of extracellular material. Collagen is produced primarily by fibroblasts, the cells found in various types of connective tissue, but also by smooth muscle cells and epithelial cells. To date, 27 distinct types of human collagen have been identified. Each collagen type is restricted to a particular location within the body, but 2 or more different types are often present together in the same extracellular matrix. Additional functional complexity is provided by mixing several collagen types within the same fiber. These heterotypic fibers are the biological equivalents of a metal alloy. It is likely that different structural and mechanical properties result from different mixtures of collagens and fibers. Although there are many differences among the members of a collagen family, all share at least 2 important structural features. Like all collagen molecules are trimers consisting of 3 polypeptide chains called alpha chains. Along at least part of their length, the 3 polypeptide chains of a collagen molecule are wound around each other to form a unique rod-like triple helix. The alpha chains of collagen molecule contain large amount of proline, and many of the proline and lysine residues are hydrolyzed following synthesis of the polypeptide. The hydroxylated amino acids are important in maintaining the stability of the triple helix by forming hydrogen bonds between component chains. Proline and lysine are important residues present in the collagen, and failure to hydroxylate collagen chains are serious consequence for the structure and function of connective tissue. It is evident from the symptoms of scurvy, a disease that results from a deficiency of vitamin C or ascorbic acid, and is characterized by inflamed gums and tooth loss, poor wound healing, brittle bones, and the weakening of the lining of blood vessels, causing internal bleeding. So that is the peculiarity of scurvy disease that happens because the collagen is not that much hydroxylated. Collagen chain is not hydroxylated, and this happens because of the deficiency of ascorbic acid or vitamin C. Scurvy disease, which causes the inflammation of gums and tooth loss, poor wound healing, brittle bones, and weakening of the lining of blood vessels, causing internal bleeding. Scurvy disease. Ascorbic acid is required as a coenzyme in the enzyme that add hydroxyl groups to the lysine and proline amino acids of collagen. A number of collagens, including type 2, 1, and 3, are described as fibrillar collagens because they assemble into rigid cable-like fibrils, which in turn become packed into thicker fibers that are typically large enough to be seen in the light microscope. The side-by-side packaging of the rows of collagen 1 molecule within a collagen fibril is depicted, and you can see it's a trimer. This cross-linking process continues to life, and many contribute to the decreased elasticity of skin and increased brittleness of bones among the elderly. Recent studies that probe the surface of collagen fibrils suggest that they are composed of strands that are twisted around the axis of the fibril to form a structure that is more like a mono-sized rope than a cable. Among the various components of the extracellular matrix, collagen molecules provide the insoluble framework that determines many of the mechanical properties of the matrix. In fact, the properties of a particular tissue can often be correlated with the three-dimensional organization of its collagen molecules. For example, tendons, which connect muscles to bones, must resist tremendous pulling force during times of muscular contraction. Tendons contain an ECM in which the collagen fibrils are aligned parallel to the long axis of the tendon and is parallel to the direction of the pulling force. The cornea is also a remarkable tissue. It must serve as a durable protective layer at the surface of the eyeball and must also be transparent so that light can pass through the lens to the retina. The thick middle layer of cornea is the stroma, which contains relatively short collagen fibrils that are organized into distinct layers. The layered structure of the stroma is similar to that of plywood. The fibrils of each layer are parallel to other fibrils in the layer but nearly perpendicular to the fibrils in the layer on either side. This plywood-like structure provides strength to this delicate tissue, while the uniformity of size and ordered packing of the fibrils minimize the scattering of incoming light rays, thereby promoting the tissue's transparency. Given their abundance and widespread distribution, it is not surprising that abnormalities in fibrillar collagen formation can lead to serious disorders. Burns or traumatic injuries to internal organs can lead to an accumulation of scar tissue, which consists largely of fibrillar collagen. A common condition described as fibrosis results from excessive production of collagen containing connective tissue. Fibrosis occurs most often in the lungs, like pulmonary fibrosis and liver cirrhosis, where the collagen containing scar tissue gradually replaces the normal tissue of the organ. Mutations in genes encoding type 1 collagen can produce osteogenesis imperfecta, a potentially lethal condition characterized by extremely fragile bones, thin skin, and weak tendons. So here we are mentioning a disease that is very important, osteogenesis imperfecta. Osteogenesis imperfecta, a potential lethal condition characterized by extremely fragile bones, thin skin, and weak tendons. Mutation genes encoding type 2 collagen alter the properties of cartilage tissue. Mutations in genes encoding type 1 collagen can produce osteogenesis imperfecta, a potentially lethal condition characterized by extremely fragile bones, thin skin, and weak tendons. Mutations in genes encoding type 2 collagen alter the properties of cartilage tissue, causing dwarfism and skeletal deformities. Mutations in a number of other collagen genes can lead to a variety of distinct but related defects in collagen matrix structure, called Ehlers-Danlos syndrome. Persons with one of the syndromes have hyperflexible joints and highly extensible skin. So, two diseases according to the mutations in type 1 and type 2 collagen is the Ehlers-Danlos syndrome and the osteogenesis imperfecta. Both consist of symptoms related to hyperflexible joints and highly extensible skin. Not all collagens form fibrils. One of the non-fibrillar collagens are type 4, whose distribution is restricted to basement membranes. Basement membranes are thin, supportive sheets, and the type of 5 collagen molecules are organized into a network that provides mechanical support and serves as a lattice for the deposition of other extracellular materials. Unlike type 1 collagen, which consists of a long uninterrupted helix, the type 4 collagen trimer contains non-helical segments interspersed along the molecules and globular domains at each end. The non-helical segments make the molecule flexible, while the globular ends serve as sites of interaction between molecules that give the complex its lattice-like characters. Mutations in type 4 collagen genes have been identified in patients with Alport syndrome, an inherited kidney disease in which the glomerular basement membrane is disrupted. So when the glomerular basement membrane which contains collagen 4 is mutated, persons get affected with Alport syndrome. Alport syndrome, that is genetic, inherited, and Alport syndrome is very important. Next one is the proteoglycan. In addition to collagen, basement membranes and other extracellular matrices typically contain large amounts of distinctive type of protein polysaccharide complex called a proteoglycan. A proteoglycan consists of a core protein molecule to which a chain of glycosaminoglycan GAGs are covalently attached. So in the case of proteoglycan, this is also seen in the basement membranes or other extracellular materials typically contain large amount of distinctive type of protein polysaccharide complexes called proteoglycan. A proteoglycan consists of a core protein molecule, then along with chains of GAGs or glycosaminoglycans which are covalently attached. Each glycosaminoglycan chain is composed of a repeating disaccharide that it has the structure ABABA, where A and B represent two different sugars. GAGs or glycosaminoglycans are highly acidic due to the presence of both sulfate and carboxyl groups attached to the sugar rings. Proteoglycans of the extracellular matrix may be assembled into gigantic complexes by linking of their core proteins to a molecule of hyaluronic acid, a non-sulfated GAG. The microscopic appearance of one of these complexes, which can occupy the volume equivalent to that of a bacterial cell. Because of the negative charges borne on the surface GAGs, proteoglycans bind huge number of cations, which in turn bind large number of water molecules. As a result, proteoglycans form a porous hydrated gel that fills the extracellular space like packing material. This property complements that of the adjacent collagen molecule, which resist pulling forces and provide a scaffold for the proteoglycans. Together, collagen and proteoglycan give cartilage and other extracellular matrices strength and resistance to deformation. The extracellular matrix of bone is also composed of collagen and proteoglycan, but it becomes hardened by impregnation with calcium phosphate salts. Both sulfated and non-sulfated GAGs are taken widely as health supplements with the aim of improving the condition of skin and joints. Next is the fibronectin. The term matrix implies a structure made up of a network of interacting components. The term has proven very apt for the extracellular matrix, which contains a number of proteins in addition to collagen and proteoglycan, which interact with one another in highly specific ways. Fibronectin, like several other proteins to be discussed in this session, consists of a linear array of distinct building blocks that give each polypeptide a modular construction. Each fibronectin polypeptide is constructed from a sequence of approximately 30 independently folding FN modules or fibronectin modules, two of which are very important. While FN type modules were first discovered in fibronectin, they are found as part of many other proteins. So this fibronectin, ranging from blood clotting factors to membrane receptors, the presence of shared segments among diverse proteins strongly suggests that many present-day genes have arisen during evolution by the fusion of pairs of separate ancestral genes. In fibronectin, the 30 or so structural modules combine to form 5 or 6 larger functional domains, and each of the 2 polypeptide chains that make up the fibronectin molecule contains binding sites for numerous components of the ECM. such as collagen, proteoglycan, and other fibronectin molecule. These binding sites facilitate interactions that link these diverse molecules into a stable interconnected network. Binding activities of fibronectin molecule are enhanced by mechanical force that pull on these fibrous proteins and unfold its component modules. Unfolding of these modules expose binding sites that would otherwise have remained buried. Binding sites for receptors on the cell surface. These binding sites hold the ECM in a stable attachment to the cell. The importance of fibronectin containing substratum for cell attachment is very important. The cultured endothelial cell is adapted to a shape unlike any it would have in the body because it has spread itself over the available surface provided by a square coat of fibronectin. The importance of fibronectin and other extracellular proteins is particularly evident during embryonic development. Development is characterized by waves of cell migration during which different cells follow different routes from one part of the embryo to another. Migrating cells are guided by proteins such as fibronectin, which are contained within the molecular landscape through which they pass. For example, the cells of the neural crust, which migrate out of the developing nervous system into virtually all parts of the embryo, transverse pathways rich in fibrils composed of interconnected fibronectin molecules. The importance of fibronectin in neural crust cell migration is readily revealed, and migrating cells are guided by proteins such as fibronectin, which are contained within the molecular landscape through which they pass. For example, cells of the neural crust, which migrate out of the developing nerve system into virtually all parts of the embryo, transverse pathways rich in fibrils composed of interconnected fibronectin molecules. The importance of fibronectin is very important. A number of organs in the body, including the salivary gland, kidney, and lung, are formed by a process of branching in which the epithelial layer becomes divided by a series of clefts. The importance of fibronectin is the formation of these clefts and salivary gland, which has been incubated with antibodies that bind up to fibronectin. Cleft formation and branching is entirely abolished as a result of the inactivation of fibronectin molecules. Next is the laminins. Laminins are a family of extracellular glycoproteins that consist of 3 different polypeptide chains linked by disulfide bonds and organized into a molecule resembling a cross with 3 short arms and 1 long arm, and they are connected into each other with disulfide bonds. At least 15 different laminins have been identified. Like fibronectin, extracellular laminins can greatly influence a cell's potential for migration, growth, and differentiation. For example, laminins play a critical role in the migration of primordial germ cells. These cells arise in the yolk sac, which is located outside the embryo itself, and then migrate by way of the bloodstream and embryonic tissues to the developing gonad, where they eventually give rise to sperm or egg. During their migration, the primordial germ cells transverse surfaces During their migration, the primordial germ cells transverse surfaces that are particularly rich in laminin. Primordial germ cells possess a cell surface protein that adheres strongly to one of the subunits of the laminin molecule. In addition to binding tightly to cell surface receptors, laminin can bind to other laminin molecules, to proteoglycans, and to other components of basement membrane. The laminin and type 4 collagen molecules of the basement membrane are thought to form separate but interconnected networks. These interwoven networks give basement membrane both strength and flexibility. In fact, basement membranes fail to form in mouse embryos that are unable to synthesize laminin, causing the death of the embryo around the time of implantation. Now let's learn about dynamic properties of the extracellular matrix. ECM can exhibit dynamic properties both in space and over time. Spatially, for example, ECM fibrils can be seen to stretch several times their normal length as they are pulled on by cells and contract when tension is relieved. Temporarily, the components of a ECM are subject to continual degradation and reconstruction. This process serves to renew the matrix to allow it to be remodeled during embryonic development or following tissue injury. Even the calcified matrix of our bones, which we think of as stable inert structure, is subject to continuous restoration. The degradation of extracellular materials along with cell surface proteins is accomplished largely by a family of zinc-containing enzymes called matrix metalloproteinases or MMPs that are either secreted matrix metalloproteinases secreted into the extracellular space or anchored to the plasma membrane. As a group, MMPs can digest nearly all of the diverse ECM components, although individual family members are limited as to the number of extracellular molecules they can attack. The physiological roles of MMPs are not well understood, but they are thought to be involved in tissue remodeling, embryonic cell migration, wound healing, and the formation of blood vessels. As might be expected from enzymes whose normal function is to destroy extracellular materials, the excessive or inappropriate activity of MMPs is likely to cause disease. In fact, MMPs have been implicated in a number of pathological conditions, including arthritis, tooth and gum disease, formation of blood clots and heart attacks, and tumor progression, etc. At least 3 inherited skeletal disorders have been traced to mutations in MMP genes, matrix metalloproteinases that are secreted into the extracellular space or anchored into the plasma membrane, which helps in the degradation of extracellular materials. Now, interaction of cells with extracellular materials. It was noted that the previous discussion that components of the ECM such as fibronectin, laminin, proteoglycan and collagen are capable of binding to receptors situated on the cell surface. The most important family of receptors that attach cells to their extracellular microenvironment is the integrins. Integrins are a family of membrane proteins found only in the animals that play a key role in integrating the extracellular intracellular environment. On the other side of the plasma membrane, integrins bind to a remarkably diverse array of molecules or ligands that are present on the extracellular environment. On the intracellular side of the membrane, integrin proteins to influence the course of events within the cell. Integrins are composed of two membrane-spanning polypeptide chains, an alpha chain and a beta chain, that are non-covalently linked. 18 different alpha subunits and 8 different beta subunits have been identified. Although more than 100 possible pairing of alpha and beta subunits could theoretically occur, only about 2 dozen different integrins have been identified on the surface of cells, each with a specific distribution within the body. Whereas many subunits occur in only a single integrin heterodimer, the beta 1 subunit is found in 12 different integrins and alpha V subunit of 5 of them. Most cells possess a variety of different integrins, and conversely, most integrins are present on a variety of different cell types. Electron microscopic observations of integrin molecules beginning in the late 1980s suggest that the two subunits are oriented so as to form a globular extracellular head connected to the membrane by a pair of elongated legs. The legs of each subunit extend through the lipid bilayer as a single membrane helix and end in a small cytoplasmic domain of about 20 to 70 amino acids. The first X-ray crystallographic structure of the extracellular portion of an integrin was published in 2001 and displayed a highly complex feature. Rather than standing upright as predicted, the integrin alpha V beta 3 was bent dramatically at the knees so that the head faces the outer surface of the plasma membrane. To understand the significance of this bent structure, we need to explore the properties of integrins. Many integrins, including the ones are given, can exist on the surface of a cell in an inactive conformation. These integrins can be activated rapidly by events within the cell that alter the conformation of the cytoplasmic domains of the integrin subunit. Changes in conformation of the cytoplasmic domains are propagated through the molecule in causing the integrin's affinity for an extracellular ligand. For example, the aggregation of platelets during blood clotting occurs only after the cytoplasmic activation of alpha 2b beta 3 integrins, which increase their affinity for fibrinogen. This type of alternation in integrin affinity triggered by changes occurring inside the cell is referred to as inside out signaling. In the absence of an inside-out signaling, the integrin remains in the inactive state, which protects the body against the formation of inappropriate blood clot. A body of evidence strongly suggests that the bent conformation of an integrin corresponds to the inactive state of the protein, one which is incapable of binding a ligand. In fact, when the alpha b beta 3 integrin containing a bound ligand is analyzed, when we analyze that, the integrin no longer exhibits the bent structure but is present instead in an upright conformation. The ligand is bounded to the head of the integrin in a cleft where the alpha and beta subunits come together. If the bent and upright structure represent the inactive and active states of an integrin respectively, then it is important to consider what type of stimulus is capable of triggering such a remarkable transformation in protein conformation. In the bent inactive conformation, the transmembrane domains of the two subunits are in close proximity, apparently held together by non-covalent interactions between the residues of the two helices. The cytoplasmic domains of integrins bind in a wide array of proteins. One of these proteins, called talin, causes separation of the alpha and beta subunits, and mutations in talin. What is talin? The cytoplasmic domains of integrins bind in a wide array of proteins called as talin. Mutations in talin that block this interaction with beta integrin subunit also prevent the activation of integrin and adhesion to the ECM. Separation of the cytoplasmic tails and transmembrane domains of an integrin that results when talin is bound is thought to send a change in conformation through the legs of the integrin, causing the molecule to assume an upright position in which the head of the protein is capable of interacting specifically with its appropriate ligand. An increase in affinity of individual integrins is often followed by clustering of the activated integrins, which greatly enhance the overall strength of the interaction between the cell surface and its extracellular ligands. Integrins have been implicated in two major types of activities: adhesions of cells to their substratum or to other cells, and transmission of signals between the external environment and the cell interior. We have already discussed the transmission of signals in inside-out direction, which primarily affects the binding properties of the integrins. Signaling can also occur in the opposite direction, the phenomenon known as outside-in signaling. The binding of the extracellular domain of an integrin to a ligand such as a fibronectin or collagen can induce a conformational change at the opposite cytoplasmic end of the integrin, especially at its beta subunit. Changes at the cytoplasmic end can in turn alter the way the integrin interacts with the host of different cytoplasmic proteins, modifying their activity. For example, outside-in signals can induce a conformational change in talin on the inside of the membrane, initiating a cascade of events leading to the polymerization of actin filaments of the cytoskeleton. Similarly, binding of integrins to an extracellular ligand can trigger the activation of cytoplasmic protein kinases such as FAK and SRC. This kinases can then phosphorylate other proteins, initiating a chain reaction. In some cases, the chain reaction leads all the way to a nucleus where a specific group of genes may be activated. Outside in signaling transmitted by integrins and other cell surface molecules can influence many aspects of cell behavior, including differentiation, motility, growth, and even the survival of the cell. The influence of integrins on cell survival is best illustrated by comparing normal and malignant cells. Most malignant cells are capable of growing while suspended in a liquid medium. Normal cells, in contrast, can only grow and divide if they are cultured in a solid substrate. If they are placed in suspension cultures, they die. Normal cells are thought to die in suspension culture because their integrins are not able to interact with extracellular substances and as a result are not able to transmit life-saving signals to the interior of the cell. When cells become malignant, their survival no longer depends on the integrin binding. The role of integrins in transmitting signals is very active area of research. Linkages between integrins and these ligands mediate adhesion between cells and their environment. Because individual cells may express a variety of different integrins on their cell surface, such cells are capable of binding to a variety of different extracellular components. Despite the apparent overlap seen in table, most integrins do appear to have unique functions. As knockout mice that lack different integrin subunits exhibit distinct phenotypes. For example, alpha 8 knockouts show kidney defects, alpha 4 knockouts exhibit heart defects, and alpha 5 knockouts display vascular defects. Explanation about integrins will be dealt in the next video.